All right, so next is Carlos. So Carlos is coming from Harvard Medical School, and he's a physiologist, and he's going to talk to us about how to link behavior and neural activity using and utilizing the neural networks. So let's hear from Carlos.
All right. You guys hear me okay? Thank you very much for the invitation. I got to start with a little question. Edgar, was that a wildcat or a domestic?
Oh, yeah. Oh, yeah, that was a wildcat.
That was a wildcat. And I guessed that because there was a little patch of green near the tail that you didn't quite crop out. But it will inspire the importance of local features, as you're about to see. So thank you very much. My name is Carlos, and I thought I turned it on. It's on. It is on. Hello, hello. All right, excellent. Let's go. Okay, so I run a visual neuroscience lab, and I specifically do this in the monkey, and I like to focus on the level of single neurons, as you can see there, and also multi-unit activity using chronically implanted microelectrodes. So if you want to understand the research program, it's actually quite simple. There's only three basic scientific premises you need to understand about this. And this is that neurons in the visual brain fire activity at different levels, at different rates. And they do this partially based on where you stimulate them in the retinotopic location of the visual field. So that actually tells you that they have special sensitivity. And the other thing is that these firing rates will depend on the kind of information that you put in that receptive field. Those are the three basic facts. Now, in the history of visual neuroscience, most of these kind of investigations of the single neuron have really relied on the subset of cell types, specifically those that encode things like orientation, direction, faces, and bodies, things that are simple, semantically interpretable, not necessarily simple as a stimulus set, but something that we can quite understand. And the study of these neurons really has given rise to this standard model of visual cortex, where you can find that V1 neurons are encoding things like lines, then V2 to V4 neurons are doing some kind of combination of lines, perhaps corners or edges, and all culminating in inferotemporal cortex and beyond, where neurons are encoding for faces, places, and bodies. And so this is a standard model, except that when we look at most neurons in the visual cortex, as we tend to do in our lab, we find that there is a lot of neurons, and in fact, I would argue that most neurons tend to have this kind of responses where they'll fire quite a lot to these kinds of image combinations, like a person, a lock, a car, a plane, or this one. You might think this is an animal-based neuron, but then you have this kind of feature. And so a lot of these neurons tend to be quite complicated. And it's kind of given us to this perspective that we tend to focus on two different kinds of neurons in the visual system. Neurons that have responses that are well aligned to some kind of simple model or semantics, the regular matter neurons, and then the neurons that are just much harder to understand, which we think of as the dark matter neurons. So when presented with this kind of stimuli and this kind of neurons, Charlie Gross, one of the early investigators of inferotemporal cortex, kind of pointed out that perhaps this meant that there is no evidence that most IT cells were selected for specific objects, and so what they were doing is basically serving in some kind of distributed population coding, which is likely right. But then it gives rise to this kind of alternative perspectives about what neurons are doing in a distributed code. In one framework, that perhaps the idea is that these neurons don't have much significance on their own. They don't encode for much. Perhaps these neurons have kind of representations that are constantly shifting and cannot be fully understood, and therefore, perhaps we should be focusing on the population response as a way to understand what the visual system is doing. And in the alternative framework, I'd like to argue, a lot of these visual neurons do have some kind of stable representation that we can kind of decode. And really the problem is that most of the time they're encoding subsemantic features that really we can't see quite easily. So what does that mean? So it means that basically the problem is that the brain really is encoding a lot of information, and only some of it is really accessible to conscious report. And I'd like to demonstrate a little bit about some of the limits of perception when it comes to understanding the relationship between these kinds of stimuli and the responses of a neuron. Now, you've all seen demonstrations of change blindness. So I'm just going to illustrate it very quickly here. This is from Chris Chamber. All you have to do is look at this movie. And we're going to present two pictures. Your job is to spot the difference between them. Just keep your gaze mostly in the center. And as you can see, these images are a little bit overwhelming, but there is in fact a change between them. Anybody see it? It's hard. With the benefit of attention, if I tell you that the pants of the gentleman right there are changing color, I think you might see that a little bit more easily. And so there I've drawn your attention to the fact that this specific change is happening, and it doesn't necessarily come to perception right away. This illustrates the fact that really it's very easy to lose track of a given feature when too many things are changing at the same time. This is one of the trade-offs that comes with perception. There's others where, for example, focusing on objects can really reduce access to local information. So this is an interesting study where people were basically asked to view an arrangement of circles. And then some of these circles were actually arranged to form kind of a Canizza square, and some of them were just set out to not do that. So they were presented first to participants, and then a second screen was shown in which the circles had just kind of moved a little bit in position, or they had changed a little bit of shape in terms of the cutouts. And then they were asked, as participants, did you see a change? And if so, what changed? And what was interesting is that when people perceived a whole, Canizza square, they were better at saying that something had changed, but not exactly what changed. And when people perceived the parts, they were better at saying what had changed, but not necessarily when it actually happened. In fact, this is true for a lot of perception, that people tend to be worse at matching certain color changes when the dots, for example, resemble something like a person in this particular example. And people are terrible sometimes at being able to match, for example, the top parts of a face, whether the same eyes, when the bottom is different. All of this to say that, that context in a natural scene can really, really change how you actually perceive the relationship between different stimuli. So here, for example, there is an actual relationship between these four images. I don't know if you guys can all see it, but this is the issue. The issue is that we're not very good at being able to tell some of these local features. So to kind of get around this problem, to solve this problem of most of these dark matter neurons, we really kind of have to have a different set of heuristics in a research program. We tend to focus on studies, we tend to avoid framing studies based on things that we can just very easily see or name. That's going to be step number one. Step number two is we tend to focus on random samples of neurons and random stimuli, no fMRI screening to kind of identify what these neurons are doing as a whole. And then we tend to rely on some supervised methods inspired by machine learning. So what I want to tell you a little bit about this is just this kind of approach, how we can use these machine learning methods for studying neurons, especially talking about feature attribution and visualization. I'm going to tell you what this has kind of guiding us about believing about the visual system, about what we're going to call the niche theory of visual representation. And then, for the spirit of this talk, I'm also going to, of this session, I'm going to highlight some specific benchmarks that might be useful when trying to find models that align better with the visual system. So let's start with feature attribution. So remember, the goal here is we're trying to understand this sort of dark matter neurons, which I think constitute the majority of visual neurons in the primate brain. And one technique that we've found to be quite useful is that of feature attribution. So this is from machine learning. It's probably quite common to most of you. This is the idea that when you're given a fully trained network in a given classification, hopefully a correct one, it is possible to then identify what features are most important. In this technique, a heat map is usually used to kind of highlight these key regions that were used for the decision. So this is a program that we started a few years ago. Here are some of my collaborators, and Victoria Zhang was the lead investigator here. The paper should be coming out in next week. But basically, the way that we do this is that we implant microelectroarrays in the brain of several monkeys, spanning from V1, V4, IT, and some monkeys' prefrontal cortex. We map the receptive fields for each one of these sites, as you can see in V1, V4, and IT. And then we have this approach where we can take a natural scene, randomly sampled, and we present it at different locations relative to the receptive field in such a way that we get the responses of these neuronal populations over the whole image. It's a way of doing weight sharing, just kind of switching it around. And what you can see is that when we look at the responses of the population over the whole scene in primary visual cortex, we find that these neurons are responding mostly to these high frequency, high contrast areas corresponding to the keyboard. And when you move over to more anterior areas like IT, the responses of the population switch more towards the hand. One thing to note, though, is that these populations really are active all the time. They're responding to the whole scene. They just tend to highlight some parts of these areas, of these scenes a little bit more than others. And what's also interesting is that, as you can see, even though we might think that ITs should be representing objects quite the same, this is an object, and yet this IT population is not responding to the object as much as to the actual hand. And this happens a lot. We tend to see all these cases where V1 responds to the entire scene. IT also responds to the scene, but focuses more on things like the monkey. And this particular population activity was consistent across most of these monkeys, particularly when focusing on animal features. So here we have this targeting of the face, a little bit of the hand. And IT, again, focuses on these three components. The hind parts tend to be also something that these neurons tend to respond to a little more. So this is all visual inspection, but we did measure the responses of these neurons over the scene by using semantic masks that were generated by other people, by participants. And if we look at the responses here of the V1 neurons inside the semantic mask and outside the semantic mask, you find that it's pretty comparable. And then as you move down the hierarchy, there's more of a response inside the mask, especially when it comes to these animal segmentations. And this actually effect didn't really hold for any mask that we came up with, especially based on objects. So it's most pronounced when we had animal masks in segmentations. Nevertheless, even though they are responding, these neurons in the ventral stream are responding more to the animal, we also wanted to know if these populations were really kind of doing a segmentation of actually taking the object and totally segmenting it in a way that you might find what some machine learning models do. So is it the case that these responses are all evenly spread around the animal, or is it more like local features? So we developed a feature-to-object ratio, which simply measures how distributed was the activity over the mask of the entire animal. And we found that, in fact, the ratio was limited only to certain parts. It almost never had a full segmentation of these neurons across the scene. And what was also interesting is that when we then asked this same question by looking at artificial neural networks, we found that, in fact, most of them differed in their similarity to the monkey brain. There were some networks that kind of already had learned somehow the same principle, things like the AT-small, the SqueezeNet, and CoreNet-S, but a lot of the same architectures kind of also failed to encode this. In fact, their performance, their ability to resemble the monkey brain, seemed to be independent of benchmarking scores or accuracy, such as brain score. So to us, this provides sort of an interesting benchmark to kind of say, if we are going to come up with some kind of model of the visual system, they should have some of these properties that we also see in the brain. Now, I mentioned that this method, this unsupervised method, allows us to find some of the local features that matter to different areas of the brain. But really, the question is which local features, and for that, we'd like to be able to focus on a second unsupervised method that Edgar has already told us about, and that's feature visualization. So, as you all know, feature visualization is a technique that generates inputs that strongly activate a model's internal units. It was developed back in 2009, perhaps earlier, and has been used a lot to understand the kinds of features, the kind of information that is learned by neurons or by hidden units in neural networks. So we decided to use a version of this using generative networks. We've been publishing about this a lot, so I'm going to go a little bit fast when it comes to the methodology. But just wanted to highlight that Binshu Wang's one of the people that have been developed very well. Binshu is sitting right at the audience, so if you have any questions, please, you can also ask them. And of course, this overlaps strongly with the kind of work that Edgar's also been doing with inception loops. But basically, in our approach, we take a sort of a model-free situation where we're going to take a generative network, which, as you know, creates images that have natural statistics, even if they're not fully photorealistic. We present them to the animal when they record from the neurons, a given neuron at a time, usually in the chronic microelectrode array. And then we use these responses and the image parameters to kind of use a search algorithm to optimize, to search for new parameters that are likely to increase the firing activity of this neuron, make it reliably active. And just a few more methods, details on the methods. The animals are just doing passive fixation. We map the receptive field, and we place the stimulus right over where the receptive field is going to be, including a little bit of the surround. And then we present the images very quickly, just once or twice. And this allows us to basically increase the activity of the neuron over a period of 15 to 20 minutes. And so what we find is that these generative networks can increase the activity of this single neurons. As you can see, though, what we get are interesting local features that don't necessarily look like photorealistic images. And even though this visualization really works in multiple areas of the brain, and here's some examples that demonstrate what they look like, what you'll find is that this explains these dark matter neurons really are not encoding objects in the classical sense, in the ways that we've been testing them. They just tend to focus on local features that may be present in objects but are not defining necessarily of objects. So one of the questions that we have is when you're presented with an image like this and I tell you that neuron is maximally activated by this, at least in that local sense, the key question is like, how could it be? This neuron hasn't really seen this kind of feature in the world. So is it possible that there's something hidden in this image? So this is why we decided to combine visualization with attribution. And the simple way that we can do that is that right after we evolve this image, we simply scramble the pixels in every part of the image and present it back to the neuron. And what this allows us to find is that some of the occlusions really lead to a major loss in the firing rate activity. And when we can see the images that really are most activating, they look like this, and the least activating are the ones that hide this one particular feature right there. And so we can actually demonstrate that it happens over the population, and then we can take that actual feature and basically excise it and transplant it to other random natural images, and we find that that can actually rescue the firing rate of the neuron, demonstrating that that local feature was necessary and partially sufficient for the responses of this neuron. And what's interesting is that even though the whole global image that I showed you looked very different from the kind of photographs that you see in the world, the local features are actually quite present in the world. So here's an example of a neuron that responded quite well when you transplanted the feature, but there were also some images that already elicited a fair amount of information from this neuron, and these are the examples of what it looked like. So basically, this neuron was looking for a local feature that this picture of a gorilla already had. So this happens to be true across a lot of the neurons that we find, that these local features that they encode tend to be present in natural images, which explains responses like this. So these features also tend to be stable over time. We've been recording from them with chronic arrays for like over a year. Binshu and Jordan have been doing work like this. And as you can see, these IT channels tend to respond, tend to encode and kind of lead to the evolution of features that, although not fully identical, tend to resemble a lot more like what they, something that is unique to that channel. So what's interesting about this, too, is that this kind of image synthesis can provide an interesting benchmark. Binshu recently decided to kind of test the ability of a neuron to drive the activity. Binshu tested whether different networks can create images that can drive the activity of these neurons using things like DipSim and Big GAN. Big GAN is a generative network that does much more photorealistic images. And Binshu found a way, basically, to interleave both generators and compete for the activation of these neurons. And found that effectively neurons in inferotemporal cortex, but not in V1 or V4, could optimize images to the same level of activity, comparable activity. And even though you get different kinds of images, the local features tend to be similar. And what's interesting is that almost no artificial neural network that we've tested, I believe none of them, can really match this ability, which tells us that these neurons have some interesting dynamics that are not part of the usual milieu of the feed-forward network. Nevertheless, what I can tell you is that over hundreds and hundreds of neuron-guided experiments, we keep seeing that most of these features tend to be local. And so that raises the question, what is the point of having an occipital-temporal hierarchy if most of what we're finding are local features, not really big objects? And I think part of the answer is something that Binshu already noted. When you actually looked across the visual hierarchy and you ask, how long does it take for a neuron to stabilize, to find the feature that it's looking for? It actually takes longer as a function of the depth, as a function of the hierarchy, suggesting that these visual features may not be getting more complex, they're just getting harder to find. So this is a way, and this is something that also happens in artificial neural networks. And so this is basically taking us to this perspective that perhaps a lot of what the visual hierarchy is doing is kind of leveraging the power of distributed coding by identifying features that are a little bit more rare. So even though the standard perspective is one where V1 neurons are encoding lines and IT neurons are encoding things like faces and places explicitly, what we're suggesting, in fact, is that most occipital-temporal cortex neurons really encode subsemantic features, that V1 neurons are simply encoding more common features like lines, and IT and prefrontal cortex neurons are encoding features that are just a little bit more complex, but still not fully representative of an object. And so this idea of a concept like an object or a face really is distributed across the whole visual network, including V1 all the way to IT. So that's what I really wanted to say for the 20 minutes of the official talk time. I had to rush a little bit, but sorry. But I really want to say that if we want to understand visual coding for the next 20 years that we have in visual neuroscience before we have a lot more AI coming in, we still have to focus on the single neuron. We have to try and understand what kind of representations they have. But I want to say that it's going to be hard to do so if we start by always beginning with our perception. Our perception, in a way, is much more limited compared to what neurons can actually do. And so it's possible that this niche tuning is going to pan out. We're going to keep looking at other areas. We've looked in prefrontal cortex. We're going to look in other regions, including the thalamus. But really, I hope I've demonstrated how sometimes taking these machine learning methods can really help us crack through some of the, the context limitations that we have in understanding visual scenes. So, yep. I just wanted to say thank you for your attention, and here's some of the work that Binshu and others have been doing, and I hope you'll find it helpful. Thank you.
Questions?
I mentioned that there was a relationship.
Session?
Yep. For the maximally excited stimulus, did you also do it for the ones which are the white matter neurons, or only did it for the dark matter neurons?
Yes, yes, we did do some recordings from face neurons in face patches. And this is work that was done by Will Chow and March Livingstone and Gabriel Kreiman. We come up with images that if you present to a person who's never seen it, they won't call it a face. But they're really good at driving the neurons. And they tend to have like eyes, and I've illustrated a couple of them. Are they face-like? Are they not? They have a certain faciness to them. But again, you probably don't want, it makes sense that they don't look like faces because face neurons also respond to things like clocks and oranges, things that happen to have some of the local features present in faces. So this, I think, helps understand that is anything a regular matter neuron. We'll see.
Okay, Dave.
It's just a follow-up question. So the sort of maximally exciting pictures they were kind of, they were presented, at least here, as static.
Yeah.
Faces are very animated. Hairbrushes are whipping up the hair. Does all this motion really drive these cells in a much stronger way or weaker way?
That's a good question. We've attempted to do work using image sequences to discover whether some of these neurons are actually more activated than others. I think it's still, I won't be able to tell you with certainty. I think some neurons can be well predicted by presenting images, for example, in a movie, at different individually, and then trying to assemble the response to the actual sequence. Some have some nonlinearities. Yeah, I wouldn't be able to say. I would be surprised if motion didn't help, if more naturalistic stimuli didn't help. But we have no evidence for that yet.
Yeah.
I'm curious about it.
There's a microphone next to
Thanks. So I have a question about sort of what it means to understand the feature selectivity of single neurons, right? Because
Yes.
let's say we map out many of them and they will all be different, and somehow it's going to be the combination of many of these single neurons that all sort of maybe funny in their own way that in the end does the computation. So even if we map out the single neuron, in the end it's going to be some population thing we have to look at. So what is that kind of description then going to look at?
See, I would have believed that until I saw Bing's talk. And the fact is demonstrated that you can start with a population description and eventually narrow down to 3 neurons that are responsible for a lot of that behavior. Kind of tells me that the old intuitions that the single neuron has a lot more information, a lot more power on the population is still true. So my interest is trying to determine what is that vocabulary. Like if you have a population response and you do need to decode it, if you're going to have a reason why this population vector is over here or not here in state space, it's going to come down to understanding what the single neuron does. So in a way, there's really no way around it, right? You can always ask why a population vector behaves like the way it does.
Yeah, I mean, so the one counterpoint that one could make is say, look, it could well be that there's, I mean, you have multiple realizability again, right? There's many different feature representations that in the end give you the same computation, even if they look different at the single neuron level.
Well, or they may not. I mean, one of the things that we need to figure out is like, are there certain motifs that tend to be represented by neurons across brains, your brain and my brain? There's certainly enough structural similarity to suggest that probably you don't have 6 billion neurons discovering 6 billion solutions and another 6 billion neurons discovering a different set of solutions. It's possible, but I think that's one of the things we need to figure out. It's a good point.
All right. Thank you very much. Let's thank Carlos one more time.